Hello and welcome to this webinar titled Designing Production Facilities of the Future, hosted by Biopharma Asia magazine and presented by Geoffrey Hodge, Chief Technical Officer at Unum Therapeutics. This presentation will be followed by Enterprise Solutions for Cell and Gene Therapy, presented by Dr. Gary M. Pigeau, Director for Center for Advanced Therapeutic Cell Technologies at GE Healthcare. My name is Stephen Edwards and I'll be your moderator. Before we begin, I would like to inform our viewers that there will be a live Q&A session at the end of both presentations. Audience members can send their questions in at any time during the webinar via the questions tab located directly below your webinar screen, and we will go through these at the end of the webinar. Now please allow me to introduce our first presenter, Geoffrey Hodge. Jeff is responsible for leading the development and execution of Unum's CMC and technical operations, including process and analytical development, manufacturing, and quality for the company's proprietary antibody-coupled T-cell receptor, or ACTR, therapies. He has over 25 years of experience in the biotechnology industry, providing bioprocessing technical solutions for companies including Millennium Pharmaceuticals, Genetics Institute, or Wyeth, and Alpha Beta Technology. Prior to joining Unum, Jeff was a co-founder of Accelerex, inventor of several of Accelerex's foundational single-use technology patents, and an operations leader at Accelerex. He holds a master's from WPI and a bachelor's degree from Colgate. I'll now be handing over to our first presenter. Welcome, Jeff.
Great. Thank you, Stephen, for that introduction and for the opportunity to share some of these ideas with the audience today. As Stephen said, I manage technical operations at Unum Therapeutics. Unum is developing a series of T-cell technologies. Actor is an antibody-coupled T-cell receptor, a T-cell engineered to express an Fc receptor and coupled to an oncology-targeted antibody. And we also have a Boxer, which is a transgene that helps T-cells to maintain activity in a solid tumor environment. My responsibility is to help bring those technologies through the clinic into commercialization. And so this presentation will be sort of from that point of view of autologous T-cell therapy. So I guess to start maybe with some high-level ideas, the problem statement is that to really design a process, you need to understand your product. That process design will influence facility design. So when we're talking about designing a facility of the future, it comes back to process understanding. The other couple principles are to create the future, you first have to imagine what the future might look like, and to imagine the future, you can learn a lot from the past and build from the past. So as everyone probably knows, it's actually close to 2 years ago now that the FDA approved the first gene therapy. It's Novartis's Kymriah, followed very quickly by Kite or Gilead's Yescarta. These are very promising therapies, very exciting. But the manufacturing times are long, in the order of 3 weeks, although they're starting to come down. And the cost is very expensive, in the order of several hundreds of thousands of dollars. So the question to the industry is, how do we take these really promising therapies and improve on the quality while also decreasing the cost and the time to make them? So one of the big challenges is that as therapies have evolved, the therapies themselves have become more complex. In the early part of last century, we had small molecules, and aspirin is an example of that, at 180 Daltons. In the later part of the last century, we kicked off biotechnology and recombinant proteins, and a monoclonal antibody is shown here that's roughly 1,000 times larger in terms of molecular weight. But it's also more complex because it's a multimeric protein with glycosylation and degradation. So it's really a family of similar molecules as opposed to a pure molecule. We're making a very similar shift now to cell therapy, which just in kind of order of size is roughly 1,000 times larger than some of the large recombinant proteins, but also is more complex in that it's not a single thing. It's a collection of cells. T-cell therapy may have other cell types in it, but even among the T cells, there are CD4s and CD8s in different ratios, and there are different levels of differentiation from naive to effector cells, so it's a very complicated mixture of cells. And coming along with that is an increasing difficulty in characterization, where HPLC might be very powerful to understand a small molecule like aspirin, and you might get a very pure peak. To understand a monoclonal antibody, HPLC trace will give you several different peaks potentially and a smear as a baseline. So we have to develop other methods to be able to characterize the monoclonal antibody. And for T-cell therapies, of course, you can't use HPLC at all. We move to something like flow cytometry and a whole series of scores of markers that give us certain indications about cells but aren't hard and fast indication of exactly the cell type. So it's a very powerful tool, but it's also at least a lot indeterminate, and so it's a very multi-parametric set of data we're trying to interpret. If we then compare autologous T-cell manufacturing to monoclonal antibodies in terms of the manufacturing process and the controls, monoclonal antibody process starts with a cell bank of a single clonal cell type and typically a master cell bank and working cell bank of hundreds of vials, so that we can start from the same place, plus or minus a little bit of biological variability, pretty well controlled. As opposed to T-cell therapies that start with patient blood, and it's a mixture of different cell types, and it's a different mix from one patient to the next. And that's also affected by the patient history, whether the patient is young or old, what previous chemotherapy they may have had, or their disease state. So it's a very different starting point. In process, productivity for recombinant proteins is based on how many cells you can grow and how much protein each cell produces. For autologous T-cell therapy, it's also how many cells you can grow up to your dose, but then it's also the viral vector transduction, or the whatever technology is being used for transduction, the efficiency of that technology. For characterization, monoclonal antibodies are typically greater than 95% pure. They're now considered well characterized. The contaminants might be immunogenic, but they're generally not biologically active at low concentrations in and of themselves. T-cell therapies, we typically get 20 to 70% transduction, so that's the active ingredient. The remainder of the T cells or other cells are sort of impurities, but they could potentially be active. T cells may, when being reintroduced to the patient, could be active in conjunction with the transduced T cells. In terms of administration, antibodies or proteins are typically an inert product in a standard IV. Cells are obviously a living product. The thawing and administration might change their activity, and then post-administration, antibodies have a pretty predictable PK. Cells are expected to expand in vivo, and that may be variable depending on the cells themselves, but also depending on the patients that they're being injected back into. And at this point in time, we're getting some clues, but it's unclear exactly what characteristics of this drug product correlate with response. So using history as a guide, the pioneering work in antibodies was done in '75 using hybridomas. 5 years later, the first patient was treated with an antibody in the US, and then 6 years later than that, the FDA approved the first antibody as therapy. Kind of a similar timeline for autologous T-cell therapies. The first pioneering work was done in '89. Took a little longer and another iteration of the CAR T technology before the first patients were treated in 2010. But 7 years as opposed to 6 years to go from that first treatment to drug approval. So a fairly similar timeline. In 2003, monoclonal antibodies were designated as well characterized, and regulation was transferred from CBER to CDER. So the question to think about is, in the similar time frame of about 2 decades, what needs to happen between 2017 to 2037 or 2034 for T-cell therapies to become well characterized? It's hard to imagine now, but I think that's where we want to try to get to. So shifting gears from there a little bit, let's look at how a manufacturing process is set up at autologous cell therapy versus a recombinant protein manufacturing facility. Typical recombinant protein facility flow starts with a working cell bank moves to a seed culture or production culture. There's a harvest and clarification step, usually some purification, a viral inactivation step, some post-viral inactivation purification, drug substance formulation, and then drug product filling. And a process flow in terms of the facility configuration, traditionally, and this is changing a bit, and I'll come back to this a little later in the talk, but a traditional facility for this type of product would be segregated by a series of unit ops or by functions. So inoculum prep separated from cell culture, separated from post-viral purification, drug substance fill and finish separated from that, and then a drug product filling separated from that, maybe even done at a different site. If you were to manufacture two different products, they would each have their own series of clean rooms that the product would move through, separated from each other. So the segregation was on the basis of clean rooms and a single process train moving from upstream to downstream. To scale this process up, you'd move from a smaller bioreactor to a larger bioreactor, and there's some art and science involved in that, but it's a pretty straightforward idea of starting at a 10-liter bioreactor, moving the same process to a 200-liter bioreactor, moving to a 2,000 or 20,000-liter bioreactor with essentially the same process flow. If we look now at autologous T-cell therapy, you can see some differences. The process flow starts with patient cells, leukapheresis. There's usually some kind of enrichment or selection step to enrich T cells, activation and expansion, transduction, expansion, cryopreservation, and then the product goes back to the clinical site for infusion. The workflow is very different. For a T cell process, typically or classically, one process would be done all within a single room, and the product might move in and out of a biosafety cabinet, in and out of an incubator, to a work surface where there might be separation devices or analytical devices, back and forth in that room. And so the room provided the unit of segregation. And in the early kind of academic numbers of patients, a perfectly feasible way to scale this up is to have a different room for each patient, wait till their batch is completed, and then sanitize that room and turn it around and have it available for another patient. As we'll talk about, that's not really as feasible for scaling up tens or hundreds of batches in parallel, and that's one of the fundamental challenges the industry faces. If you look at what those two types of manufacturing layouts mean when comparing autologous cell therapy to recombinant proteins, recombinant proteins generally have more of a challenge in terms of material flow and waste because each room has to have its own material staged and waste removed. Same with personnel and process flow. People have to go in and gown up and gown out of individual rooms per batch. A very similar problem in terms of environmental control. The HVAC in a recombinant protein facility, you can flow the air from clean to dirty along a single process line and make more efficient use of HVAC. For a autologous T-cell facility, each room needs to have a segregated HVAC to prevent cross-contamination of patient samples. Each individual room needs to be cleaned, and there needs to be an environmental monitoring program for each room. So there's considerably more burden for that segregated room-based process flow in terms of control of environment. The little bit of good news is from a building-wide utilities standpoint, autologous T-cell processes, because they're bench scale, tend to be a little gentler in terms of utility demands. You don't need clean steam or purified water in most cases. You can purchase these things as pre-made buffers and sterilized. The bottom line here is that the batch time and labor issues really get amplified in an autologous T-cell process. Because you can't stockpile drug the way you can with recombinant proteins, the manufacturing process starts when the patient is leukapheresed, and it only ends when they receive their drug. And that lead time between leukapheresis and infusion is critical to the patient, and that's what the industry needs to try to shorten. Because of that, the release time is critical. With an antibody product, you can have a batch on the shelf while you're making a next batch and releasing it and replacing your inventory. Here, the patient waits until the batch is released, until they can receive their treatment. In terms of process understanding, as I said earlier, we have very limited process understanding. It's still somewhat an academic process, and it's very labor-intensive, requiring a lot of manipulations. And the release testing not only is time-consuming, but it becomes a significant percentage of the batch labor, where releasing a batch of antibodies the cost might be distributed over thousands or tens of thousands of doses. The cost here is batch per batch. So every batch goes through the same entire release process. And the release testing is very equivalent to recombinant proteins. It's not a lighter load. All of the safety testing is relevant, and in fact, there are a few more safety tests. And the same types of characterization tests are expected for autologous T-cell therapies. So how do we go from there from where we are today to advancing the technology and where we'd like to be in the future? So first of all, don't underestimate the importance of incremental improvements over time. So on the left is the first mobile phone, the Motorola Dynatac from 1983. It weighed 2.5 pounds, and it cost about $4,000 in the day, which would be about $10,000 today. In last year, you could buy from the same company, Motorola Moto X, weighs about 6 ounces and costs under $200. How did we get from the left to the right? It wasn't a single invention or a couple big inventions that moved us there. Phones got smaller and lighter. The screens had more and more functionality, and to the point where we got touch screens and phones with apps. And in case you think that might be unique to kind of hardware and computer technology, the same things happened in the antibody or recombinant protein industry, where the average monoclonal antibody titer has increased incrementally year after year, and not from any particular innovation, from improvements in cell lines, in expression vectors, in growth media, in batch feeds, in bioreactor techniques. All of these things have been incrementally improved to the point where we've seen essentially a log order of improvement, where we're getting tens of mgs of antibody per liter in the 80s to hundreds of mgs in the 90s to single-digit grams per liter earlier this century. And as we're coming to the close of this decade, people are starting to push 10 grams per liter for some of the systems. So the point there is that new technology and breakthroughs are only part of the answer. They're unpredictable, and they're driven by understanding the product, which we're in the very early stages of. So there probably will be some breakthroughs, but they aren't the total solution, and in fact, they may not be necessary. Incremental improvements in existing technology stacked up over time may get us a large part of the way there. Learning how to use existing technology more efficiently, and also making sure the regulatory pathways are sufficient to guarantee product quality, as they must be, but also purpose fit for T-cell therapies and not trying to mash a round peg into a square hole. So now I want to shift gears again a little bit and look at some of the technology that currently is enabling and some of what could be enabling in the near future. I think a lot of people who have started off in the autologous cell therapy field take single-use systems for granted. They are absolutely enabling. This field would be very, very difficult if it weren't for single-use systems. But single-use is a fairly new idea to the field, to biotech in general. Back just a couple decades ago, it was a radical idea to manufacture a recombinant protein in single-use systems. Nowadays, you'd be hard-pressed to find a company that isn't incorporating single-use at least into some part of their GMP manufacturing. And the reasons for this are pretty obvious. You can eliminate the need for cleaning and sterilization and the need to validate those functions. You minimize or eliminate the chance for cross-contamination by throwing away what's exposed to the product, so that it's not carried over to the next batch. And there are some real operational costs. The capital cost, at least for an antibody facility, is roughly 50% of what it would be if you built that same facility in stainless steel, and the space required is maybe a third to a half the size. And there's also an operational cost savings that I didn't list here that's usually anywhere from break even to about a 30% savings. So single use has been a major benefit for recombinant proteins. It's the default because of the need to guarantee no cross-contamination. It's the default for autologous cell therapy. But there's a benefit that's yet to be realized, and that is the ability to close up systems. So single-use systems are very amenable to being closed. First of all, you can customize tubing sets and bag sets and equipment to address some of your specific product needs. But there are also a variety of connection technologies, tube welders and aseptic connectors that can be validated to show that they maintain a sterile connection and a sterile fluid path. So if you were to do this, there's yet another advantage of being able to close up a system, and that is the ability to start to declassify your room and do multiple products in the same room and declassify your air handling requirement. And I've got an example here. On the left, you can see surgeons performing open heart surgery, and that has to happen in a place where the room has been sanitized, where the patient is scrubbed down and covered, where the people performing the operation are using sterile gloves and masks and covered as much as possible. The environment, the threat of the environment has to be mitigated by controlling the environment. On the right, you've got kidney dialysis, which is happening in a much less controlled room. Why is that? It's because kidney dialysis, by the nature of the process, has been designed to be minimally invasive. The patient doesn't have to be opened up and exposed to the environment, so the risk is much lower. If we can do the same thing with a biomanufacturing process, that should allow us to declassify rooms and design multi-product facilities. And just to illustrate how powerful this could be to the industry, here are a couple case studies. The first is a replacement of Class A with isolator. This is a specific company that did this work and decreased their Class C space. They calculated about $1 million a year savings for a reasonably small manufacturing facility of 3,600 square feet. About half of that cost savings was from a reduction in environmental monitoring, and the additional savings was from reduced utilities, labor, and gowning and cleaning supplies. So pretty significant savings in that operation. The second case study is an industry survey of hypothetically what would the benefit to your process be if you could go from Class C space or ISO 7 space, if you speak that language, to controlled non-classified space. And the average was not quite a 50% reduction in capital costs and a greater than 50% reduction in operational costs. So you can imagine if we could apply this to T-cell therapies, that this could have a very significant cost impact to these therapies. So another technology that is, the technology exists but not yet in a state where we can use it across the board is rapid analytical technology, very much in need for autologous cell therapy. There are rapid microbial methods available, and some companies have validated these to replace the compendial 2-week sterility assay. There's also promise of PCR and next-generation sequencing techniques that could potentially allow us to consolidate the assays for multiple adventitious agents into one test, reduce the sample size required for that test, reduce the testing timelines, and replace the recombinant retrovirus or lentivirus testing, which right now is a very long and expensive and cumbersome cell-based assay. So DNA or nucleic acid techniques have the potential to be able to replace a lot of assays. There's an FDA working group and a number of technology groups working on this, and I think it's got great promise for the future, although it's not quite there yet. We also need improvements in cell potency assays and functional assays, and as we come to understand our products better, I'm sure these will develop. Especially, we need markers for cell proliferation. It's known that these cells do need to proliferate and survive in the patient to have a long-term effect. What's not known exactly is how to assay this in vitro and what translates from an in vitro assay to clinical efficacy. Some more enabling technology, again, things that exist now but will need to be further developed for our field, is automation and IT. So certainly automation of unit operations has the advantage of being able to reduce human labor required and therefore reduce expense. It can also reduce human error, and that's something that is often overlooked. Sometimes human error can result in a lost batch, and of course in this case that would mean a patient that doesn't get treatment. More often though, deviations are minor and the batch may be suitable for use, but it does result in a lot of human effort to go through and do investigations and close deviations, and that's a lot of extra cost and labor to assure quality that might be avoided through automation. Similarly, electronic batch records and manufacturing execution systems can help reduce labor and help reduce human labor, or human error, rather, and have the potential to be able to reduce the amount of content that QA has to review to be able to release a batch by validating fields or accepting or showing that certain input has been validated to be correct. Then humans have to look at a smaller data set. And then finally, as we look toward the future of this type of therapy, big data analytics and machine learning, this is a very multi-parametric space. So it's hard for a human being with an Excel spreadsheet to find critical differences when there are many, many different variables that could be interacting. So certainly machine learning and big data analytics will help us to understand this complex therapy. Just some things to think about when considering automation and some common mistakes. Don't automate too early. Once you automate a process, it's harder to change it than it is with a manual process. So if you automate a process too early and it's still changing frequently, you may end up spending more effort than if you finalize or mature a manual process and then try to automate it. Along similar lines, don't expect automation to fix a poorly designed process just by throwing IT at it. You need to make sure your process is manageable and working well first. For example, if you've got an approval process that requires a 10-page justification and 5 levels of signature on paper, and you try to automate that, you haven't fixed the fundamental problem. You need to remove some levels of approval and streamline the justification. A computer's not going to help you with that. And then finally, design the process for automation from the beginning. There are a lot of examples of processes that work well with humans that people then try to design automation to do exactly what the humans did, when there might actually be a different way to run the process that would be more amenable for a machine to do it. And if you can think of that in the beginning, you can build that into your process. So now let's look at where the industry is and how to apply some of these ideas. Right now, there are 2 competing visions for how to do autologous cell therapy manufacturing. One is what I'm calling the distributed ops, which is basically you look for each instrument that is best at doing a particular part of your process. You buy all of those different instruments, and then you figure out how to connect them together, both physically and electronically. You have to figure out how to make and break tubing connections between these, and you have to figure out how to make electronic connections between these. That has the advantage of being very flexible. You can switch equipment in and out if it breaks or if a better technology comes along, but it's got the disadvantage of not being designed to be connected. The opposite of this is the integrated type of system where everything's been designed to be connected and work together up front. That minimizes that connectivity problem, but then you're dependent on a single source supplier because these are generally proprietary pre-made units. If a component fails, you lose the entire unit, and you can't swap out for better technology in one particular part of the process. Right now, scale-up is really essentially doubling down on one or the other of these visions. People are taking the distributed unit ops and trying to figure out how to run multiple bioreactors with one piece of critical equipment and kind of debottleneck the equipment utilization, but it still doesn't solve the fundamental problem of connectivity. Or there are companies building modular cassettes that are designed to plug into a kind of a switchboard for control, and they help solve the setup problem, but you're still working with a very restrictive single process at a particular volume and particular process flow with particular analytics that have to be built in advance and dependent on a single vendor. So we've been looking at a third way. We're at for the moment calling ACTIA for autologous cell therapy industrial automation. The idea here is to think of a different way of going about manufacturing, which is to take all of the functionality out of the cell culture vessel and put it in the hardware. So start with something very simple, very low cost, like a gas permeable bag. Use robotics to move those culture vessels from incubators, which is where they'll spend 95% of their time in a kind of a low budget temperature control operation, move them out to a series of analytical or separation workstations to do cell counting, add or subtract media, add viral vector, wash, etc., and then put those cells back into an incubator. Overlay this all with process control. And the idea here would be to build an integrated system that has the benefits of automation, but allows the flexibility to swap in or out different workstations as they break, or to configure for a new process, or as new technology comes along. And so I kind of already gave away this slide. We think this has kind of the best of both worlds. It has the scalability that you get and the automatability that you get from a multiplex system, but it gives you the flexibility that you get from a distributed unit ops system. And there is some precedent for this, and maybe starting from right to left, there's a technology that people may be familiar with to take tea flasks and culture cells in tea flasks and passage them and trypsinize them. So basically this is a brute force method of taking an inefficient human process of culturing cells by T cells and putting a lot of work into automation system to mimic what a human being does. Very successfully, but the process wasn't really designed to be automated. It was a machine imitating what a human does. In the middle, there are multiplex bioreactor systems. These are aseptic as opposed to sterile, but show that you can run culture control and monitor cultures for a lot of different cultures in parallel. And then maybe most relevant, moving to the left, this is a system similar to something we have at Unum, where within a HEPA enclosure, we have a robot that has a robotic arm in between an incubator and a liquid handler and a centrifuge and a plate reader, so we can incubate plates most of the time. We can move them out and spin them down and change media, or we can not spin them down and pull samples to do cell counts on the imager, and or pull samples out for other assays. And we've run cultures out to a month long in this aseptic environment. So what we're really looking to do from a GMP setting is, instead of aseptic, translate that to a sterile manufacturing operation. And there are a few enabling pieces of technology that we're working on to try to get there. Custom designed culture vessel that's specifically designed to be amenable to operation as opposed to be amenable to humans, although usable by humans. A sterile connection and liquid transfer device. And then there are a couple pieces of technology that I think we can take off the shelf but customize: integration and scheduling software and multiplex separation technology. So pulling all that together into what I hope will be the facility of the future is hopefully we won't even need a controlled classified space, but to the extent that there may be a transition phase, we may need a small classified space to stage liquids or components to allow closed system operation then in a central kind of ballroom environment with workstations dedicated to, restricted around equipment pods with incubators around them, arranged around limiting process equipment with electronic batch records and automation to move both physical samples and data back and forth with automated or semi-automated workflows, eventually statistical process control and machine learning. A QC lab that has automated assays wherever possible, or even online or at-line assays in the manufacturing process, and then hopefully a smaller QA function that has to review less data because some of the data analysis from QC has been automated and some of the process control and cross-checking from the manufacturing process has been automated. So what do we need to get there? First of all, there's a lot we can learn from the recombinant protein industry in terms of running a process, clean room design, analytics, and facility ideas. But there are also some things we have to unlearn because our industry is currently very good at scaling up, but we're not very good at doing lots of things in parallel. So there are other industries we may need to look at for inspiration and ideas for automation and logistics and tracking and building out in parallel. We need collaboration among cell therapy companies and vendors to develop better methods for product characterization, better process equipment, components and controls, components to enable closed processing, for example. We need good, robust product tracking and logistics systems. And we need cooperation between industry and regulatory agencies to make sure that while we're ensuring sufficient product quality, we're not overburdening the industry and we're enabling new technology and new innovation. So with that, I'd like to thank you and turn things back to Stephen.
Thank you for that great presentation, Jeff. Now, before I introduce our next presenter, Dr. Pigeau, once again, I would like to remind our viewers that there will be a live question and answer session at the end of the webinar. Please feel free to send in your questions at any time during the webinar via the Questions tab located directly below your webinar screen, and we'll go through these at the end of the presentation. Now, please allow me to introduce our second speaker, Dr. Gary Pigeau. Gary Pigeau received his Ph.D. in biotechnology from Brock University in St. Catharines, Ontario. He completed a postdoctoral fellowship at the University of Alberta with a visiting scientist appointment at the University of Oxford. Gary moved to private industry in 2008, where his research group focused on process development and scale-up of a proprietary bioprocess technology. He then moved to commercial, large-scale biomanufacturing in 2013. Gary joined GE Healthcare and the Center for Advanced Therapeutic Cell Technologies, or CATCT, at CCRM in 2016, and led the pluripotent stem cell process development, scale-up and manufacturing efforts. Today, Gary is a GEHC director of the CATCT, delivering on customer-focused fee-for-service process, media and assay development projects, development of new products for GE Healthcare, and internal technology development projects to continue to develop and grow capabilities within the group. I'll now be handing over to our second presenter. Welcome, Dr. Pigeau.
Thank you, Stephen, and thank you to Jeff for a great presentation and laying a lot of foundational information for what I'll present to you today. I think you'll find a lot of alignment between the two talks today. So I'm speaking to you today from the Center for Advanced Therapeutic Cell Technology in Toronto, Ontario, Canada, where we have a collaboration with CCRM, the Center for Commercialization of Regenerative Medicine. We will be talking around facilities of the future, so here are some forward-looking statements as we are talking about a crystal ball. Things may change as we develop these technologies. So GE Healthcare is a global solution provider spanning the industry in many, many ways. Today we'll be focusing on solutions in the life sciences business, and in particular in the cell and gene therapy business, where we are working on addressing challenges in developing workflows for manufacturing these transformational therapies. So we look at this graphic which describes the dynamics of cell and gene therapy manufacturing, and the time axis here is a little bit arbitrary, but regardless, we are on the left-hand side at this point. We are making the 3-pound Motorola phone in cell and gene therapy where the processes are highly manual. We've only just had the first commercial launches, and the tools and the processes that we use to make these therapeutic products really are just developing, or we're borrowing them from bioprocess, or we're inventing them as we go. So this is meant to illustrate how we see the industry develop as we increase exponentially the patients treated. We see the cost and the risk of manufacturing dose decreasing, eventually culminating with what we hope to be the end of the clean room, or at least the declassification of certain manufacturing spaces, and really the reduction of the manufacturing to closed and automated systems, which I'll speak more around today. So here we have a timeline of some key events in GE Healthcare's evolution as we've advanced technology in cell and gene therapy. I want to draw out some specific examples that really give the sort of three key tenets of the talk today. So for developing facilities of the future, in my opinion, the first thing you need is the manufacturing process development. We need a process to start. It can be manual, but the idea being it should be evolving towards closed and automated. So circled in green here at the end of 2015 is the partnership between GE Healthcare and CCRM, where we do perform process development and process closure that we'll talk about in a little more detail. The next bit, the second part, is, I think, the integration of a digital overlay for manufacturing. This has been an ongoing issue or focus in GE Healthcare. Obviously, we are an industrial digital company. I haven't circled the Venetti point here in 2016, but the Chronicle event in 2019 really is the first dedicated manufacturing automation solution that GE has been able to provide. So this highlights the need for that second pillar in the facility of the future, being the digital layer for manufacturing. And the third being the facility. So we've got three bullets here on the timeline dating back to 2014, where the QBio product was first deployed in bioprocess. We'll talk more about QBio today. This is a modular, pre-built, prefabricated solution for manufacturing, and of course it started out in bioprocess with monoclonal antibodies. The first cell therapy dedicated QBio unit was manufactured in 2018, in the middle of our timeline here, and then more recently, in January of this year, we announced a collaboration with GCON to provide modular, prefabricated clean room pods, which are available for installation into a pre-existing warehouse type environment, and we'll talk more about that today. So the three parts that we'll get into are the process development, and as you might imagine, we'll talk about CAR T manufacturing today. The integration of the digital layer and what that means for manufacturing, and then the actual facilities to house the manufacturing workflows themselves. So starting out with our process development efforts in collaboration with the Center for Commercialization of Regenerative Medicine, we entered into a collaboration 3 years ago here at the Toronto site, surrounded by hospitals and universities and in the middle of what we call the Discovery District. The intent of this collaboration is to develop the next generation of manufacturing workflows for cell and gene therapy, with the end goal of making these more accessible to patients. The facility is on the in the Discovery District with over 100 people contributing different expertise. The process development group is where the GE folks are focused, but we also have, for example, GMP manufacturing available on site with 8 clean rooms and 2 clean rooms dedicated to vectors. So GE Healthcare is a tools provider. We manufacture hardware and applications for manufacturing across many different industries. What you see on this slide is where GE Healthcare fits in to the immuno-oncology manufacturing for CAR T products. We make equipment for thawing apheresis units, for isolation. We do not have hardware in the activation space. We do, however, have hardware for the remaining steps, with gene transfer, expansion in the jury, or if you're from bioprocess, the wave platform. Harvest can be done in a couple of different units, either the CPACs or the Cephia, as well as formulation and the subsequent cryopreservation in the via freeze platform, which I won't go into too much detail about that, but it is a really interesting product that does not require liquid nitrogen to freeze your cells. And then, of course, reformulation with the CPACs. We have several things in development from those future forward-looking statements outlined at the bottom in development with stem cell technologies out of Vancouver, British Columbia, and those we expect to commercialize in the future. So it's great. We have all of these pieces of hardware, and it looks like they can play very important parts in the distributed unit ops that Jeff was talking about. But how do we bring it all together? And that sort of comes back to the group at CCRM in Toronto. So one of the projects that they worked on was taking this process and working towards automating it, closing it, and integrating these unit ops. So the workflow here runs from top to bottom, with the apheresis coming in at the beginning of the process and a cryopreserved product as the output at the end of the process. The legacy process was open, manual, and disconnected. A lot of work in biosafety cabinets with flasks and manual additions. And the workflow that's been designed here is indicated in the darker gray, which is a closed workflow. Yes, lots of tube welding, but the idea being that this is something where we could present to a regulatory body and demonstrate that we perhaps don't need a clean room per dose, because these are closed, connected, and automated instruments. We want to provide the solution to challenge the manufacturing paradigm, and that's what we've done here. And if we go back, at least thinking about the gap that we had in selection from our hardware perspective, you'll notice on this slide on the second unit op on the far right, we have a Clinimax inserted in this workflow because that was the automated solution that was available for this process. So it makes sense to build the process for the intended workflow with the best hardware that we could have available to us. So that's what we've done. And we put this together, and you can find this white paper on the GE website describing the closed and semi-automated processing of CAR-T cells for cell and gene therapy. So that is really sort of the first part. That is the idea of, first we need a process if we're going to think about how do we integrate into a facility of the future. The second part has to do with the digital overlay, and for that we have been developing a manufacturing automation system named Chronicle. And Chronicle essentially sits over top of all of these unit ops. They can be GE hardware, they can be third-party equipment, but the idea is that the machine data from all of these unit ops is collected into one central cloud-based server. This then leads to the creation of electronic batch records. And you can see illustrated on the right side of the slide, this is a electronic SOP. So the idea being for an operator in a GMP facility, in order to progress through the workflow, again, we want to get paper out of the clean rooms. So the idea is we can follow the procedure here. These tablets are equipped with cameras, so we can talk about barcoding and linking reagents to the products as part of the electronic batch record, which is really important. The actual manufacturing data that's generated from the equipment, again, is appended to that batch record, and all of that can then be displayed on a heads-up display like we see on the left here, essentially visualizing what your facility might look at any given point in time. The idea being that we need a solution that takes the art out of the science of manufacturing. And this is part of the solution in that regard, is to advance our manual manufacturing to something that's more automated and looking like an actual commercial manufacturing protocol. This is an example of some control data this is from an actual T-cell expansion. This is in a jury, and we have the actual machine data shown in blue, the average of that data running in green, and then the control ranges in red. And the downward deflections that are occurring every day are the operator opening the lid to the instrument and withdrawing a sample. This is an example of the machine data that we collect, but we probably don't look at nearly enough to be considered automation in manufacturing. So this is a control chart that shows on, it looks like maybe the 78th hour, that the temperature decreased beyond that lower control point. And for this example, let's think about the idea that temperature is absolutely imperative for your product. So we have the data, we're tracking the data, now we have the opportunity to look at this point and decide if this is a deviation that will keep the product out of a patient. Conversely, if this deviation was not central to a critical quality attribute, this could then be an opportunity for training the workforce, because it looks like at this point, again, the lid was open for too long, so the operator possibly spent more time than was needed withdrawing a sample, or there was a problem withdrawing the sample. The point is, if we're not interrogating the data that comes from these machines, we're not doing the process a good service. So the idea that invariant data streams coming from the processes represent candidates for control plots and definition of the process. There's a second class of data that comes out of these manufacturing workflows, and that's the data that changes with time. The variant data streams that are changing in response to the metabolism inside the vessel. These cells are growing, they consume glucose, they consume oxygen, they release lactate, they change the pH. There's a lot of dynamic sensor-based data that we can get, and what I'm showing here is some work that's in development, but the idea being that from these variant data streams, such as oxygen demand and gas flows and pH, we can use that to correlate to our cell counts. And the impact here, or the desire being that if we can enable a inference, a soft sensor of the cell counts, then we don't have to take a sample from a CAR T manufacturing process. And this is really important because every sample point represents cells that don't go to the patient. It also presents an opportunity for contamination that we'd like to avoid. So in addition to decreasing the risk in the process, it allows us to start thinking about how do we integrate these unit ops. So if we take that idea of being able to predict when these cells would hit a target cell count for the patient, we can use that to start to stage our downstream and indeed stage administration of the therapy to the patients. So this slide depicts a potential dashboard with 3 juries expanding cells in the center. And the idea of this predictive cell growth on the right here saying, these cells will be ready in 3 days or 2 days or 3 hours. And the implications of how this rolls into the operation of the facility, the scheduling of the labor, the scheduling of the unit ops that are downstream, these are all things that we're just starting to really wrap our head around. How do we make the facility of the future run as such? So this is the last slide on the digital part, showing that dashboard sort of live in a good manufacturing practice suite, where the operators are able to quickly look at the status and make real-time process decisions. So the last part of the talk will be to discuss the facilities that will actually house these manufacturing workflows for cell and gene therapies. First, we'll start with the QBio offering from GE Healthcare, which is a modular prefabricated solution, which is, again, it's turnkey. It's designed to be assembled, so there is a reduction in the amount of build time for a facility. It is meant to be deployed on a greenfield site. It's a standalone structure that can be configurable, but the intent here is that it's much faster than a greenfield build, which through the course of development of your therapy would allow for a deferral of that investment. So this slide shows sort of the progression across the top from IND to launch of a therapy, and when you might need to build a facility to manufacture your product. Because of the time it would take to build a traditional facility, you may need to think about building this in early phase 2, late phase 2. Because QBio is modular and has a quicker time to deploy, you could consider this closer to the end of phase 3 and mitigate the risk of failure through your clinical trial. So there's some advantages there with respect to the development pathway, in addition to the capital benefit as well. The idea that these are pre-built and uniform means that if we're going to deploy facilities around the world, that they will be the same. Again, we want to make sure that we're manufacturing these products in a uniform manner so that the impacts are consistent, and really the way to do that is to think about the way in which they're manufactured, and the facility is a big part of that. Next slide shows some of the key events in getting time to market. The idea being that with the prefabricated solution, that we can address many of these things in parallel. And so here we have an estimate of between 1.5 to 2 years from start to finish, from the initial planning stages all the way to deployment of the QBio facility in your choice of location. I mentioned at the beginning that this started out with a history in bioprocess, and this is not simply an idea. This has actually been deployed. There is a monoclonal antibody QBio in China, and you can actually go on YouTube and search for this, and you can see a time-lapse video of these modules being manufactured and transported on a truck. They're actually manufactured in Germany in this case. They're transported to a port where they're loaded onto a cargo ship and shipped to their destination in China. They were assembled, and within 18 to 20 months, these are probably hard to see, but the data on the bottom left shows the increase in titer as the facility got up and running. So what we've done is we've taken what we've learned from deploying the QBio product in the monoclonal antibody space, and we've taken that to cell and gene therapy. We're saying here QBio goes viral, but also not only for manufacturing the virus required for CAR T, but also for manufacturing the CAR T cells themselves. So this is meant to describe QBio sort of reimagined fit for purpose for cell and gene therapy, and to have the flexibility expected from these sorts of solutions. This is very much similar to the earlier slide, where it's an expandable and it's a turnkey facility. The difference being that it is biosafety level 2, which is required for manufacturing of cell and gene therapy. And the idea, I'll show you some workflows and how the people and the products flow through a schematic of the facility, but they need to be unidirectional to be GMP compliant. You can't have the waste crossing over with your raw materials, and so you need to take that into account when we design the layout of the facility. And the intent for these facilities is to manufacture virus, vectors, as well as CAR Ts. So there's 3 versions of that shown here with sort of expanding footprints. The standard facility is just over 2,000 square meters. and then goes all the way up to almost 4,000 square meters with the doubling of that. The idea being that one could build now and expand later, or if the market was defined and you wanted to build the large facility up front, that would be a possibility as well. But we often talk with people around, how many doses per year could you support, and then what happens when this takes off, because we saw the exponential graph from Jeff's presentation where these technologies tend to lag a little bit, and then all of a sudden the industry booms. This is meant to address that, where we have the ability to expand the QBio offering. Thinking about the GMP compliance for workflows, we can see on the left, this is the sort of the materials flow, where the raw material comes into the facility in the green lines and enters the clean rooms from the bottom of the graphic. The waste is segregated and goes out of the on the orange or red lines, and then the product goes through QC and then eventually to release. And again, the people flow is important to consider when designing these, especially when the facility gets quite long and you're working to map out the travel of the product and the people. Maybe putting a QC lab on the very distal part of the facility is not the best idea, because that really isolates some of the rooms. So we spend a lot of time thinking about how to lay these out in a way that makes sense for manufacturing down the road. The layouts and the different grade areas are indicated on this slide, where the actual upstream and downstream production areas are shown sort of in the center in the darkest red as grade C or ISO 7. This is the biosafety level 2. The supporting labs are as well, because materials travel to those labs for analysis and qualification there. The incoming materials, we need to include areas for media and buffer preparation, but also for kitting out these systems. We talked a little bit about the game changer that single-use technology was. That's something to keep in mind when designing a facility like this, is that you have to have a place to essentially lay out the single-use consumables and inspect it for integrity and make sure that it's ready before it goes into manufacturing. It's something that needs to be considered on design of these facilities. So this is sort of how it's been laid out, and this is that starter package. The additional sort of the number 2 and number 3, that adds on more manufacturing space, where you would keep your sort of media and buffer prep areas as well as your utilities and your office, and only expand on bona fide manufacturing space to make the most of that expansion. The last part that addresses the manufacturing environment, the facility itself, is the recent collaboration with GCON. This will feel very familiar to what we've just seen with QBio, because it has at its heart a very similar concept of prefabricated modular facilities. And so GCON, there's some information here on the company, and they have some, again, some good YouTube clips here where they actually show you an installation of a pod in less than a minute. It's actually a very it's an interesting one to watch, because they slide these pods around on air skids, and it looks really neat to watch them install it. So in January at Facilitate, we made the announcement that we would be collaborating with GCON. This is the press release here, and the idea being that we have QBio, and it is designed for a greenfield deployment that gets to market and gets to manufacturing faster than a stick build, if that makes sense. The second part here with the GCON is perhaps there's a facility that's already existing and they need to add clean room capabilities. You could add these onto a QBio, or you could add these onto a facility that is already in operation that requires more capacity. So the partnership here is that GE will provide we call the flex factory equipment, all the equipment that we started at the beginning of this talk on the sort of CAR-T manufacturing workflow with the distributed unit ops. we would provide that inside a pod where a cell and gene therapy provider may want to manufacture, that this would be a deploy solution, which would be sort of rapid deployment to market. If a supporting facility, so this picture here on the left shows that it's actually inside a pre-existing warehouse, and that's one of the things for to have the options of how we deploy the manufacturing facilities of the future. So, and this is just sort of the layout. It's probably a little bit tough to see, but the top down here on the left showing sort of those segregated areas like Jeff was talking about. And in this facility, these are in the center of that top down, you see sort of 2 rows of 4. That's meant to describe 4 juries or 4 waves, expanding cells, with the idea that, again, that part of the process would not need to be completely segregated into a clean room, but I think that that is still a really good conversation to have with the regulatory bodies in the specific regions in which you wish to deploy. It doesn't make sense from a manufacturing perspective for one dose to take up a full clean room, but that, again, that's outside of our ability. What we want to do is provide the engineering and administrative solutions that really challenge that paradigm. So I think this is almost my last slide. The idea being the cell therapy manufacturing facilities of the future are not just the facilities. They have to have the appropriate hardware and processes. I'm a firm believer in the digital layer with statistical process control as well as predictive analytics that really will enable this real mature cell therapy manufacturing. The manufacturing automation solution that we provide, again, all mature biologic manufacturing have some sort of overriding MES system. So the idea being this one is built for purpose for cell and gene therapy. And further on the built for purpose idea, the modular facilities are a way to get to market quicker, perhaps to maximize footprint that has already been established with the GCON, and really to get these therapies to market much, much quicker. And I think that is my disclaimer slides. So thank you for tuning in today, and thank you, Stephen, for the invitation.
Thank you, Dr. Pigeau. Now we will begin the question and answer segment of the webinar, but once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question is, what is the biggest challenge in manufacturing autologous cell therapies? And I believe that's for you, Dr. Jeff.
I think that the biggest challenge is really that we're very early on in understanding these products, and so the processes are evolving, the types of equipment that vendors are supplying is evolving, and they're very manual early stage processes, so that contributes to a very high cost of manufacture and a long turnaround time.
Thank you. Our next question is, as technology changes, will these current offerings be able to accommodate advancements? And that's for you, Dr. Pigeau.
Thank you. I think so. I think that was the idea behind the expandability and the flexibility of these processes, the idea of process definition. These are really universal. They're not limited to autologous T-cell manufacturing. I spent a lot of time in the allogeneic world, and we didn't touch on that today, but the idea being that cell and gene therapy manufacturing, it will change, and we have a responsibility to be ready to adapt.
Thank you. Our next question is, when should people start planning their facilities? Again, that's for you, Dr. Pigeau.
That's an interesting one. You might think the answer is straight away, but I don't think it is. I think that planning the facility really depends, of course, on the process, but I think once you understand how many doses you might want to manufacture, or in the allogeneic world, what the maximum volumes you might want to process might be, those are sort of the key decision points that will directly impact the design of your facility.
Moving on, our next question is, what is the biggest barrier to decreasing cost of goods for autologous cell therapies? And that's for you, Jeff.
You know, it's actually kind of a similar answer to the first answer. I don't think there's a single big barrier. There are a lot of barriers, but they're all driven by the fact that we're very early on in our product understanding, and so the processes and the equipment and in fact the facility designs are evolving.
Not a problem. Our next question is, how will automation change autologous cell therapy manufacturing? Again, that's for you, Jeff.
Yeah, so I think it will really have a significant impact on autologous cell therapy manufacturing, maybe just not quite yet. Certainly companies are moving toward applying automation selectively, and there will be some incremental benefits. Before I think we can get to the vision of a very highly automated facility, there needs to be a little bit of invention, both in terms of equipment and enabling technology, but also some innovation in terms of facility design, regulatory path, and process design. So I think as people start to see the potential for automation, process development scientists will start to design processes to be more automatable, and gradually we'll get to a point where you can imagine the entire process being automated.
I could add a little bit into that T-cell manufacturing workflow. I can think of a couple of examples where, for example, a feed rate is increased when a certain cell density is achieved. If we think about what I was showing today on the ability to predict what that cell density might be, then it follows that you could cascade that control to increase the feed rate of the pump driving the feed. That's a really simple example that would take out a manual intervention from the process, and I think we're just getting to the point now of learning how to automatically turn those knobs, if I'm going to use the right words.
Great, thank you both. Our next question is, how will digital enable scalability? And that's for you, Dr. Pigeau.
Sure. I think it comes down to process definition. I think the idea of listening to your process and collecting all of the machine data that is available is the raw material for scaling up your process. If you can't define your process as completely as possible, you really don't have much chance of scaling up.
Okay, our next question is, if it is a closed system, can we use the same room for multiple cell process? Jeff, would you like to answer that first?
Yeah. I mean, I think I addressed that. You can. There's some work you may need to do to demonstrate that it's a closed system, but recombinant cell protein company or recombinant protein manufacturing companies are already doing this. So it's not uncommon to have multiple bioreactors for multiple products in the same room now.
And great. Do you have anything to add, Dr. Pigeau?
Obviously, that's something that we're sort of working towards, but my favorite regulatory answer is it depends. It really does depend on the environment, and I don't mean the environment of manufacturing. I mean the regulatory environment that you're dealing with, and having those open conversations with the regulators around your process, and showing them, as Jeff mentioned, that this is what we've designed, this is where we think it's safe to go, and again, that collaboration with the regulators is key to getting this into a ballroom style type of manufacturing.
Great. Our next question is, aseptic closed system connection is a great point for our industry. What kind of testing do we need to do to prove the regulatory bodies or to the industry to decrease grade levels in operation space? Jeff, would you like to answer this first as well?
Yeah, sure. And kind of go back to Gary's point. It depends on the regulatory body you're talking to. I mean, the fact is that most regulatory agencies are willing to see data. They don't tell you yes or no that you can or can't do something. They say, if you want to do this, here are our concerns, and then you need to generate the data to alleviate their concerns. So if you're going to say, we're going to close a process and eliminate classification of our clean room or decrease classification, the burden's on you to generate a package of data to show that your process really is closed and what that means to you and how you know that it can be reliably operated as a closed system. So, so for example, people have done this with tube welders where they've done spore challenges, coated the outside of tubes with spores, done a number of welds, passed media back and forth through that welded connection, and shown that the media remains sterile. So you can validate these connection technologies, and you can validate a process as being functionally closed, and that should provide the regulators some data and some confidence to believe that you don't need to rely on a more or less clean room to protect the product quality, which, if you think about it, is kind of a primitive way of assuring product quality, hoping that there aren't too many germs in your room, as opposed to completely closing the process. I think we'll look back on that as a very old-fashioned way of doing manufacturing.
Dr. Pigeau, do you have anything you'd like to add?
I've had similar thoughts around what you might do to demonstrate that a tube weld is indeed a sterile connection, and there is not a prescriptive assay to do that. I had thought about, just you weld tubes together, you run clean media through it, and you submerge the weld in a saturated liquid of E. coli, and you show that your pass-through liquid has no contamination. That's an example, but that's just out of my head. That's not prescriptive by any means. But if you were going to approach the regulators with this conversation, again, you would need to go away and come back with some sort of data that supports the idea that indeed this is safe.
Great. Moving on, our next question is there any effort focused on working with clinicians to ensure more consistent, high-quality cell preps and leukopacks that come out of the clinic to help make the cell and gene therapy manufacturing process more robust? Again, I believe that one is for Jeff, so if you'd like to answer that first.
Yeah, sure. So there are efforts. A number of companies have done some work in this space, and we're among them. There are also a few consortia and collaborative groups looking at trying to standardize leukapheresis practices. That said, leukapheresis, there's a whole other presentation. In fact, one of my colleagues is giving a workshop at an upcoming seminar on this topic. So it could be a whole separate topic. But leukapheresis technology doesn't really purify cells. It enriches white blood cell fraction and decreases the number of red blood cells and the amount of plasma. But it's not really a purification technology. So you can only get so much out of it. So there may be some things that companies want to do to try to control it, but don't think of it as a purification step that can be tightly controlled to give very pure product of different conformation based on the process you run.
Great, thank you. Our next question is, do you think the investment of automation for multiplexing processes will be fully utilized if capacity varies? Would the cost of the facility be too burdensome, but do you provide the justification, the ROI for such systems? Dr. Pigeau, would you like to answer this first?
Yeah, I lost you a little bit, Stephen, near the end, but I think I've got most of it. And the idea, I think, is we invest in this facility, we invest in the automation, but maybe it's not being utilized completely. And how do we manage the return on investment? Is that fair?
Yeah, I think we can go with that.
Okay. Well, the idea being, and we've worked through some of these scenarios as with ramp-ups in mind for automation and for manufacturing throughput, and that is being thought about because when we talk about the aspirations, you want to do 2,000 autologous doses per year. What happens when that becomes 10,000? And what happens if I open the facility tomorrow and I actually only need 15? How do we rationally scale up the manufacturing hardware facility and digital layer? I think it's doable. I think that there are some creative financing solutions required for investing in a new industry. But we have been working on ramping models for these so that it's not, we talk about being ready for commercial production quickly, but once they're up and running, the idea being that you ramp production based on demand.
Yeah, if I could add to that, I think it's incumbent on the manufacturing companies to build a strategy that ends with automation but doesn't necessarily have to start with automation. So, and that's one of the points in my talk was to think about automation and how to design systems that are automatable, but that doesn't mean you have to invest millions of dollars in automation to make your first batch of product. You can design systems that human beings can manipulate such that when you go to automate them, there will be minimal change to those processes. And so you can design physical manual processes with automation in the future. Then you can selectively start to introduce automation where it makes sense for your stage in clinical development and your level of investment. So you don't have to go from 0 to 100% automation. You can stage it over time. But to be able to do that in a way that makes sense, that doesn't require you to tear things out and replace them, you have to know where you want to end up first. And then you can build it in pieces and pull it all together at the end.
Great, thank you both. Our next question, and I believe this will be our last question looking at that time, there is a lot of talk about closed systems and sterile versus aseptic. However, in the presentations, aseptic welders and aseptic single-use connectors are being treated as closed systems. Is it really viewed this way from a regulatory perspective for room classification versus bags pre-connected and sterilized together, i.e. a true closed systems? Dr. Pigeau, would you like to answer this first?
Yeah, I think we were in this space a little while ago. The idea being that if you are looking at sort of the integrated unit ops, this is currently how we need to approach it. If you are in an all-in-one type solution, and that is the advantage of that, it takes away the flexibility and it gives you back the idea of a truly closed system. These sort of sterile welds, they are the current state-of-the-art for integrating unit ops, and we've designed workflows that leverage that. And the idea being that is the current state-of-the-art that a cell therapy manufacturer would take to a regulatory body and propose as a manufacturing workflow.
Maybe I'm reading a little into the question, but the question specifically talks about aseptic welders and aseptic single-use connectors. Right. And of course, aseptic is not sterile. So I think you have to be careful with what the vendor is capable of claiming versus what the technology has the potential to do. So the vendors may not have done all the work to claim that they're sterile welders or sterile connectors, but the manufacturing companies can put that work in. The analogy I've used is a stainless steel bioreactor. Companies do not sell sterile reactors, but of course, it's expected that you will be able to sterilize the bioreactor you need to. It's incumbent on the manufacturing company to do the media holds and develop the sterilization protocols and develop the data package to show that they can sterilize a stainless steel bioreactor. Same thing for tube welders and aseptic connectors. Not to say that you can't use them as sterile connections, but there's some amount of work that you need to do to prove that to the regulatory agencies.
Great. Thank you for your answers, and thank you, audience members, for your questions. Before I finish the webinar, I would like to ask our presenters if they have any closing remarks. So, Jeff, do you have any closing remarks for our audience?
Nothing major. It's just I'm very excited about the potential of this field. I think cell therapies have established themselves as a third modality that I think have a really incredible future, and I'm very excited about them as a therapy. But from my particular field, I'm also very excited to make them faster and more efficient and more affordable for people.
And Dr. Pigeau, do you have any closing remarks for our audience?
Maybe just a note of thanks for the opportunity to join you today and to hear Jeff's presentation. And to any of our listeners and participants today, if you are interested in further developing your process or any of the commercial offerings, you can find me through LinkedIn and we can continue the conversation separately. Thank you so much.
And I would like to take this opportunity to thank our presenters. Once again, that was Geoffrey Hodge, Chief Technical Officer at Unum Therapeutics, and Dr. Gary M. Pigeau, Director at the Center for Advanced Therapeutic Cell Technologies at GE Healthcare. Thank you for sharing your knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers back to you. Thank you, everyone. Have a great day.